Ladies and gentlemen, can we begin? Thank you. Nice crowd for the last night. Thank you for coming. I want to start with an apology. Lots of people who read the brochure said to me, you're supposed to do, I think, bacteria last night, or was it the somatic marker hypothesis? Gut-brain axis. Well, I'm really sorry about that. You know, I prepared these lectures rather late and found myself having too much information and I had to adjust. So I'm sorry.
Come back next week.
I will. And during the December holidays, when I was expected to have a holiday, I found myself in the position of the Vice-Chancellor of the university. And it wasn't a quiet time, so I never had a chance. I never had a chance. So I'm sorry about that. And I want to just say to you that tonight you'll have to stop me at some point because I have a lot of information to go through. So let's begin. Thank you. Now, I want to start with something, this book, Descartes' Error by Antonio Damasio, who's a neuroscientist and who's written some excellent books on the brain and the mind. And in this book, he asks us to visualize 3 scenarios. The first one is the thought experiment we did here the other night. Can you all hear me?
Yeah.
The thought.
Can you raise the volume?
I hope there's no echo. Okay. Is that all right? Better? Better, sir? Better? It's an echo. Do you want to come to the front? Yeah, come to the front. So the first one is similar to the thought experiment we did the other night, where I said I'll keep you here until kingdom come and your blood sugar levels fall. Now Damasio says in a situation like that, which is pretty common, your sugar levels fall, but you're not consciously aware of them. Ultimately, your consciousness comes in when you realize you're hungry. But he says here, the process involves no overt knowledge, no explicit display of options and consequences, and no conscious mechanism of inference up to the point when you become aware of being hungry. So that's the first scenario. The second one is, imagine you're sitting there and I throw a cricket ball at you and it's heading straight for you. And of course, you see it coming and the first thing that comes to mind is you need to protect yourself, so you will try and duck the ball. Now, that doesn't give you a chance to start reasoning or thinking logically or saying, now, must I dodge? Which way must I dodge? Must I dodge to the left or to the right, or must I bend over? Those things don't happen. You do what you have to do to protect yourself from being hit by the ball. That, again, doesn't require very much logic or reasoning. The third scenario is when you're placed with a number of options or problems. Who should I marry? Or if you're a judge, how should I judge this case? Or if you're a mathematician, you're trying to teach a difficult concept to a class, and you think about it. So that's overtly involving logic, reasoning, thinking about it, rationality. Now, the 3 scenarios look different. 2 of them are quite, basic. And the third one is what Damasio might call higher order thinking. Now, this is where he feels that Descartes made an error, because Descartes, of the 17th century, said, I think, therefore I am.
Je pense, donc je suis.
Okay. So Descartes gave birth to the idea of dualism, that we have a mind and a body. And I want to talk a little bit about that dualism, but it's not dissimilar to the point that I showed you the other night about acid. Somebody in 1925 said no acid, no ulcer, and sent humanity on a certain trajectory to deal with ulceration. Now, here's a man in the 17th century, a very clever man who says, I think, therefore I am. He has separated the so-called mind from the body. Now, Damasio says that's an error. And he says that to make a final response of those scenarios, you must apply reasoning, and that involves holding a great many facts in your mind, telling results of the hypothetical actions, and matching them against intermediate and ultimate goals. So based on the blatant differences between the third illustration and the former two, it is not surprising to discover that people generally assume that one and the other have entirely unrelated mechanisms, mentally and neurally, so separate indeed that Descartes placed one outside the body as a hallmark of the human spirit, what we know as mind, while the other remained inside, the hallmark of animal spirits. Now he thinks that that was an error. So, what he says is the point I want to stress is that your mind is not a blank at the start of the reasoning process. Now, if any of you heard of the evolutionary psychologist Steven Pinker, who wrote The Blank Slate, where he said there are no noble savages. All of us come hardwired with a lot of our behavior. So, rather it is replete with a diverse repertoire. He's talking about the brain or the mind. Rather, it is replete with a diverse repertoire of images generated to the tune of the situation you are facing, entering and existing your consciousness in a show too rich for you to encompass. So, how do we deal with a difficult situation? So, you are a business person, and somebody makes you an offer. Hey, I've got this fantastic deal for you. And you say, oh, this is what I've been waiting for. This is going to be fantastic, and I'm going to be rich. The only problem is that the person who made you the offer is your best friend's worst enemy. Right? And now you have to make a decision. The decision is, I want to enrich myself, I want to be comfortable in my life, I want to look after my family. And then there's the other possibility that I will lose a very good friend who will tell me, how dare you associate with such ... Okay? So now, you have a decision to make. Now, in your mind, you start thinking of all the possibilities. How will my friend take it? What if my friend breaks off with me? We won't be able to go fishing on Sundays. It'll change my life because we've been going fishing for years and years. That kind of thing. So all those things come to mind, and when the bad images come to mind, you feel a certain uneasiness, not in your head or in your brain, but in your soma or in your body, most likely in your stomach. Now this is so obvious, but a scientist had to actually break it down for us and say, that is your somatic marker hypothesis. And he says the advantage of that is that it accelerates a decision. It helps you choose the best possible scenario from the rest. So that is somatic marker hypothesis. And for me, that was very exciting because it also relates. Because I've had that experience so many times, and it also relates to what the modern neuroscientists are saying. They're saying that our emotions, our feelings, are all physiological responses. Now, if you see what I've got here, this is a diagram I got from Damasio's other book, Consciousness, the feeling of what happens. He talks about the relatively simple stereotype patterns of response which include metabolic regulation of what we were talking about. So you can't actually de-link whatever is going on in your physical body from what is going on in your mind. So from the physical body, you get the basic life regulation. It happens in you, it happens in your cat. Then you get emotions, primary, secondary, tertiary emotions, right, which are complex stereotype patterns of response which includes secondary emotions, primary emotions, and background emotions. Emotions give rise to feelings, which are sensory images signaling pain, pleasure, and emotions become images. And that image, when you become aware of it, you become conscious of it. And we are conscious being. And from there you can go on to high reason. But link this whole thing. It's one process, and they all physiological processes. Do you notice something about this diagram? There's no spirit, mind, or soul. It's all material. So this is what I mean when we talk about materialism or physicalism, which is quite in opposite to what Descartes said. Okay, thank you. I'm glad that's over. Now, people still write about this. Complex and hidden brain in the gut makes stomach aches and butterflies. This is an article by Sandra Blakeslee in the New York Times. Sandra Blakeslee has written a book with one of the world's greatest neuroscientists, Ramachandran, from San Diego. And the book was called Phantoms in the Brain. If any of you, very much like Oliver Sacks, but with more neuroscience in it than Oliver Sacks ever had. So in this article, she says, the reason for common experiences, scientists say, is that the body has two brains, the familiar one encased in the skull, and a lesser known but vitally important one found in the human gut. Like Siamese twins, the two brains are connected. When one gets upset, the other does too. Okay, so here's some big people. Thomas R. Insel, who's the director of the National Institute of Microbiological Health. Much of what makes us human depends on microbial activity. Bacteria in the gut produce vitamins. They help break down our food. Their presence or absence has been linked to obesity and various diseases, such as inflammatory bowel diseases, and the toxic side effects of prescription drugs. It also has an enormous implications for the sense of self. He's an American neuroscientist and psychiatrist, and he should know, because he's also the Director Institute of Mental Health in the USA. Right? Now, you get harmful and harmless gut bacteria. So, and there's an explosion of research in this area. But how do we distinguish between harmful and harmless? This is what it looks like in your gut. You're not clean at all. Okay, and these bacteria are small. Simon Carding says, this is on the tip of a pen. Yeah, that's the tip of a pen. And the tip is 20 microns across, and the bacteria are 5 microns long, so they can all fit in very nicely on the pen. And you get millions and millions of them on the tip of a pen, so that's how small they are. But this diagram you won't be able to see, but I'll just tell you what it says, that 1 in 10 cells in the body is human. Our microbiome, in our bodies, human cells are outnumbered 10 to 1 by bacteria. Some 8 million genes function in this invisible universe, more than 300 times the number of our own cells. And what they've done is they've taken every part of the gut and counted the number of species of bacteria. So let's see, the tongue has 7,947 species. The inner elbows have 2,012. The vaginal opening, 2,062. And it goes, so it goes. And the large intestine has 33,000 species of bacteria. So we're all bugged out. Now, just forgive me, I'm sort of repeating 3 or 4 slides that I might have used before in the first talk, just so that those people who weren't here can just see this. So we have 10 trillion human cells. That's what we're made of. We have 100 trillion microbial cells. 1 linear centimeter of the colon carries more bacteria, 100 billion, than all the humans who have ever been. So we are more bacteria than human. Now, the most prolifically microbe-rich organ is the large intestine, which in the average American is home to a dynamic ecosystem of 1,200 different bacterial species. These creatures, collectively known as the microbiome, produce a slew of compounds, from toxins that cause inflammation and are associated with heightened heart disease, risk to critical vitamins, the mood-regulating hormone serotonin, and compounds that suppress hunger and reduce glucose and insulin levels in the blood. Now, we spoke about harmful bacteria the other day. We spoke about Helicobacter pylori in the stomach, and we're not going to go back there. But there's another bacteria which, if you have an overload of E. coli, can land you in hospital. It's a very dangerous bacteria. But now, the idea of a relationship between bacteria and our cells is old. It's very old. The Earth is 4.6 billion years old. And a simple cell, just a cell, the first form of life, is 3.4 billion years old, right? Now, a complex eukaryote, that means the cells that we have. So what happened is, first we had simple cells, bacteria and archaea, and then we had these complex eukaryotes that are only 2 billion years old. But what made them complex is they developed a nucleus and they developed something else. They had a visitor. And the visitor was a bacteria. And that bacterium wasn't welcome into the cell 2 billion years ago. So the cell asked it to get out, and the bacterium said, look, if you let me stay, I'll do you a good turn. I'll be your powerhouse. Lucky it didn't say I'll be your ESCOM, because that wouldn't have worked. I'll be your powerhouse. And that bacterium today is known as the mitochondria. Right? So it's resided in the cell for 2 billion years, and what a powerhouse it is. This is where glucose is finally, by the process of respiration, makes ATP. Remember I told you about ATP? If you come back next year, I'll tell you all about it. Okay. So that was a very successful symbiotic relationship. That means there was mutual benefit to both the cell, the host cell, and the bacterium, which we know as the mitochondria. Somebody who worked out this complex event of respiration. We think respiration is breathing in and breathing out. No, 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 no. That oxygen has to get in right here. And that's where the action takes place. So, somebody in 1959 by the name of Mitchell won the Nobel Prize for this, and justifiably too. But you can read this book by Nick Lane from University College London, Power, Sex and Suicide, Mitochondria and the Meaning of Life. And he gives you the entire history of this bacterium. So you see, you've come to listen to me about the gut, and I'm telling you what's the meaning of life. You didn't expect this. I'm just a very nice guy. Okay, so the gut is the inner tube of life, as you know. It carries microbes, the location and identification of which are in progress. Ladies and gentlemen, this is a brand new field. We don't know much about it. We don't even know all the species there, and we haven't distinguished between all the harmful species and the beneficial species. So it's new. Gut microbes and what we eat and who controls who. It's a good question. Gut microbe therapy, Simon Carding says, is like lawn care. Okay? Now, I told you about the mucus meeting I go to once every 2 years. It's an international meeting. And this one last year was the 13th one. So it was our 25th anniversary. So we celebrated there. And over there was Natalie Juga, my friend from Norwich, who works at this institute, Institute of Food Research in Norwich, and she presented this paper. It was the first time at this Congress, after 25 years, that somebody, it's a mucus conference, it was the first time that somebody presented a paper on the microbiota and their relationship with mucus. So she says the mucus layer covering the tract plays a key role in maintaining a homeostatic relationship with our gut microbiota. Okay, and then she says it's an emerging paradigm, and the mucins act as the food for some of these bacterial species. And in this talk, she gave insights into how gut bacteria adapt to the mucosal environment, focusing on number one, mucin utilization, okay, by one mucin degrader strain. So there is one species there, Ruminococcus navus, that actually feeds on the mucus. And there's another one that just adheres to the mucus. It's called Lactobacillus reuteri. Okay? And she went on and spoke about this. She gave a brilliant talk. She's an excellent scientist, and she works with Simon Carding. And she told me after that, Anwar, I didn't know you were interested in microbiota. Simon Carding will share his talk with you. I'll speak to him about it. And that's how this idea came about, that I should give the summer school lecture, because Simon Carding did share some of his slides with me, and I'm very grateful for that. And others I sort of photographed off the screen, so they didn't come out very clearly, but I'll explain them. And then Malin Johansson from Sweden, the University of Gothenburg, she spoke about intestinal mucus dynamics, and she said the mucus organization, which is linked to its function, is influenced by the local extracellular milieu determined by ion transport. So she is looking at another aspect of the relationship of microbes and mucus in the small intestine. And then Eric Martins gave a talk on colonic mucins by symbiotic human gut bacteria during health and disease. So there were 3 mucus and bacteria papers. Now, if you remember Mark Light, I spoke to him about the other night. He collects monkey poo, and it's, he says in the gut we have 2.7 kilograms of bacteria, which can form an organ. They have 2 million genes compared to over 25,000. The gut microbes communicate with the nervous system using the same neurochemicals that relay messages in the brain. You wouldn't believe what we're extracting out of poo. Now, this is Mike Lyot, and he initiated the Human Microbiome Project in 2007. He studied a list of psychoactive compounds in the feces of infant monkeys. His hypothesis was that gut microbes communicate with the nervous system using some of the same neurochemicals that relay messages in the brain. I mean, this is the shocker. Because we always think that everything happens in the brain. So you think of a neurotransmitter, it's got to be in the brain. Somebody's depressed, oh, they've got too little of the neurotransmitter. What's going on? But it's always the brain. And here he's saying your microbes make neurochemicals. Okay? He then wants to transfer microbes from one monkey to another to see how the neurodevelopment is affected. The advantage over invasive neurosurgery. So it is an advantage. Instead of having neurosurgery, why not just have a sort of microbial transplantation? And what a suitable alternative to even attempt at altering someone's genes to change behavior or cause a disease. Can you imagine what a revolution this would be in medicine? And you're the first in Cape Town to hear about it. Fantastic. Okay, so I did some reading around the field when I did have some time. And Mark Light had a paper in PLOS Pathogens called Microbial Endocrinology in the Microbiome Gut-Brain Axis, How Bacterial Production and Utilization of Neurochemicals Influence Behavior. And he says that microbial pathogens in the brain cause behavioral changes, right? Now, if you have micropathogens in the brain, that means you're sick, you're ill, because they don't belong there. Okay. But the microbiome in the gut influences behavior in a non-invasive manner through the production of neurochemicals that are identical to those in the central nervous system. Perhaps we need to modulate the gut-brain axis to treat behavioral anomalies. Look how far we've come. From here, now we're actually studying this. Simon Carding's paper, I suspect, which was in Microbial Ecology in Health and Disease in this year, Dysbiosis of the Gut Microbiota and Disease, I think is going to be the definitive paper in this field. And what happens is other scientists read it, they get ideas from it, and then they develop the field. So he says, first let's find out what a healthy colonic microbiota is. What is the best population of microbiota that each one of us needs in our gut to be healthy? So there is a pivotal mutualistic mutualistic relationship between colonic microbiota, their metabolic products, and the host immune system. The bacterial species residing within the mucus layer of the colon, either in direct contact with cells lining the epithelium or in indirect contact through their metabolites, will influence whether host cellular homeostasis is maintained or inflammatory mechanisms are triggered. So that relationship between the microbiota and the mucus is particularly important. There is evidence that perturbations in the gut microbiota involved with the development of colon cancer. So don't upset the population. High protein diets can produce carcinogenic metabolites from colonic microbiota that cause cancer. I've read this paper, he said nothing about the Banting. Microbiota produce neurosignalers and neurotransmitters. GI disease may be a comorbidity to central nervous system disease and psychological systems. Could we develop new probiotics to treat depression? You're going to say yes. Okay. So, dysbiosis, also called dysbacteriosis, is a term for a microbial imbalance on or inside the body. The dysbiosis is most commonly reported as a condition in the digestive tract. Dysbiosis of gut microbiota is associated with pathogenesis both in the gut and elsewhere. Gut-related disorders include inflammatory bowel disease, IBS and celiac disease, which is hypersensitivity to gluten. Outside the gut, you get allergy, asthma, metabolic syndrome, cardiovascular disease, and obesity. So you interfere with the population. And this is what is worrying these doctors, that we use antibiotics left, right, and center. And what are we doing to our gut population? So then there was this third paper. So I chose only three, by Evrancel and Seeland. And they say this is on the gut-brain axis, the missing link in depression. The gut microbiota is essential to human health and the immune system and plays a major role in the bidirectional communication between the gut and the brain. Evidence that the gut microbiota is associated with metabolic disorders such as obesity and diabetes and neuropsychiatric disorders such as schizophrenia, autistic disorders, anxiety, and major depression. Microbiota may be involved in the development of brain systems. So we're going right down now to the fetus and the development of the brain. Imagine microbiota being involved in that. Early research shows that probiotics have an antidepressive and anti-anxiety effect. Okay. So, gut microbes and hunger hormones that act on the brain. It says probiotic bacteria can raise tryptophan plasma levels and reduce hunger-inducing hormones in the hypothalamus. The commensal bacteria, the ones that live happily in your body, can produce mimics of peptides regulating satiety and hunger, like leptin, ghrelin, PYY, and NPY. So, I mean, this is how far this is going. It's beginning to embrace the entire biochemistry of your body. Gut bacteria can influence eating behavior. Directly, peptide mimics of hormones, indirectly by stimulating autoantibody production that block appetite regulation. Okay? Now,
Should we all be taking probiotics?
no, hang on. Okay, now, this is, what's her name? Sonia Shah. Sonia Shah wrote an article in the New York Times recently called The Diet Myth. It was written on December the 28th, 2015. I was working, where were you? Okay. The Good Gut and the Hidden Half of Nature. Now, the title was The Diet Myth, but she says there's sometimes before, here, just read that. Now, what I want to tell you now is I showed you 3 research papers. I showed you 3 abstracts presented at the Cambridge meeting. So what I'm saying now is that there's an explosion of research taking place in this area. Now there are books, and you are aware of 2 books that I showed you, which were This one is very recent and this one is a little older. But now, just recently, last month, 3 new books came out on the subject. So she says, sometime before the age of 5 or so, my son misheard the term taste buds and imagined instead that his mouth was alive with taste bugs. This came in handy for him when under fire for rejecting the variety of new foods we put on the table. It wasn't his fault that he didn't like broccoli and spinach. He'd explain it was his pesky taste bugs. So the first book is by Tim Spector. Tim Spector wrote The Diet Myth, Why the Secret of Health and Weight Loss is Already in Your Gut. Then there was another book called The Good Gut, written by Erika and Justin Sonnenberg. They say, take control of your weight, your mood, and your long-term health. I'm beginning to wonder why we have a brain. The bugs are doing everything for us. And then David Montgomery and Ann Bickler say, the hidden half of nature, the microbial roots of life and health. Sonia Shah is a science journalist and a prize-winning author. Okay? So this was the picture in the article, taste bugs or taste buds. She says, we were exasperated, but it turns out that my 5-year-old was right. Popular and scientific interest is how the microbiome might be manipulated through diet, supplements, and transplants have exploded in recent years. The microbiome produces a slew of compounds from toxins that can cause heart disease to critical, and also critical vitamins, serotonin, and hunger suppressants that reduce glucose and insulin levels in the blood. Wipe the microbiota before the age of 2. Wipe it out with antibiotics and the risk of obesity jumps by 11%. Antibiotics in livestock is responsible for childhood obesity, says Tim Spector. And Tim Spector did a very interesting experiment on obesity, and I'll tell you about it just now. So, Sonia Shah says, using the improved detection capacity of genetic sequencing techniques, scientists have discovered that 100 trillion microscopic creatures live in and out on the body, influencing everything from the intensity of our immune responses and our moods to our dietary preferences and propensity to gain weight. So what do the new books say? That gut microbes are neither good nor bad. The part they play in health and disease is dynamic and contextual, Tim Spector says. Then the Sonnenbergs say that gut microbes are the good guys and they're under assault. Western lifestyle diminishes the diversity of the microbiome. Processed foods starve gut microbes of nutrients. Highly sanitized environments deprive them of microbial visitors. Antibiotics knock them down. Caesarean sections distort their populations. By doing so, they argue these hallmarks of the Western lifestyle are disastrous, and partaking of them is tantamount to a game of Russian roulette with our health. The Good Gut is the book that they wrote. And The Hidden Half of Nature, and this I got from the reviews, by the way, the microbial roots of life and health. The excitement about the microbiome is undoubtedly justified. Its discovery calls into question all manner of underlying assumptions about the nature of disease, our relationship to the natural world, and even what it means to be human. And then here is the popular press. I mean, there's articles now coming out. I don't think there's a New York Times in any week nowadays that doesn't have an article about microbiota. And you get when gut bacteria changes, gut brain function, autism's gut-brain connection, the gut-brain axis, the diet myth. Should we bank our own stool? That's a good one. And complex and hidden brain in the gut makes stomachs and butterflies. So, the rich array, Peter Andre Smith says, of microbiota in our intestines can tell us more than you might think. So the brain-gut cooperation begins early in life. Now this is from Julia Enders. Brain-gut interaction is responsible for the emotional world of the baby. Consider the pleasant feeling of the full stomach. discomfort of the hunger pang or the wind, a child's relief after being burped. Adults rely on their senses, but the brain-gut connection remains. Bad mood or well-being are gut feelings. In 2013, they found that a cocktail of certain bacteria altered the emotional and pain processing parts of the brain. It's very hard to believe. Now, there's some experiments that I have to mention to you. They're not comfortable, especially if you're sensitive about animal experiments, but I have to give them because this is what's going on. So there's a question: how intensely are we prepared to strive for something that we believe exists? Something concrete like getting to dry land, high school graduation, or something abstract like satisfaction or happiness. So research into motivation and depression has been done in a mouse model. The mouse was in water deep enough not to reach the bottom. How long will it be able to swim for? So the stressed mouse with depressive tendencies gives in. They get better with antidepressants. But John Cryan fed mice with Lactobacillus rhamnosus, JB1, a strain of bacteria known to be good for the gut, and the mice swam longer, were more motivated, and had fewer stress hormones in the blood. Performed better in memory and learning tests than the groups not fed bacteria. Sever the vagus nerve, and there's no difference between the groups. It's all happening in the gut. There's another experiment. The gut is responsible for feelings, gut decisions, and behavior. Stephen Collins, he said timid and docile mice of a Balpsy strain, and exploratory and gregarious mice. So he had two groups. He had a docile group and he had a gregarious group. He treated with antibiotics to wipe out all the microbiota, and then he interchanged bacterial populations for the two groups. They swapped behaviors. So the docile ones became gregarious and vice versa. Not tried in humans yet. Okay. But Simon Carding says gut microbes can transfer emotions. Here's a timid mouse. You give him a fecal transplant and he becomes a happy mouse, right? And you take an adventurous mouse and you give him a transplant from the timid one, and the germ-free mouse is not so happy. So what happened here? So the timid mouse, he has feces transplant and he becomes a happy mouse. So he becomes bold. The adventurous mouse has a transplant, and the germ-free mouse, if he's given the same feces from the adventurous mouse, he becomes very happy. There. So, you remember this one? The wholesale replacement of gut microbes. Okay, doc, let me see if I've got this right. You want to put shit up my ass to cure my diarrhea and my addictions and allergies. But as Simon Carding says, it works in treating gut infection and works very well. It's more effective than antibiotics in the treatment of C. difficile infection, 94% cure rate. You won't believe what we're extracting out of poop. This is Mike Light. Our gut contains vast amounts of these microbacteria, and we only have 20 to 25,000 genes, and of course we need sugar to keep them alive, and there are 2 million unique bacterial genes in each microbiome, and from the point of view of DNA, we are more bacterial than human. But how do these gut bacteria affect the brain? So microorganisms in the gut secrete substances similar to the ones used by neurons to communicate and regulate mood. Serotonin, dopamine, and GABA. These play a role in intestinal disorders which coincide with anxiety and depression. Certain bacteria in feces is associated with depression. Now, I just put this diagram down. This is a nerve cell or neuron, okay? And can you see that neuron has a head, right? And then there's dendrites, so it's receiving information there. So that's the body and that's the axon. And the axon then will go on and have these here, right? Can you see that? And that will join on to the next neuron. So when you have a nerve impulse, it moves that way and then comes here, and then it joins up with other nerves and sends it. So if it's a thought, or happy thought or whatever. Sorry. But I wanted to show you a common reflex arc, right? So here it is. Here's a receptor. I put my hand on a hot stove. The message gets to here, and then so it's from the peripheral nervous system. Here's my hand on the hot stove. So this is the receptor. I have a sensory nerve that will take it right down to the central nervous system, perhaps the spinal cord or the brain. Now it transfers that information to an interneuron, which then goes out there again to the other neuron, which is now an efferent neuron, and it says, hey, ask this dude to move his hand from the stove, he is getting burnt. It all happens in a split second. But what I wanted to show you is, do you notice that these little projections here, dendrites or whatever you call them, they don't actually touch. Can you see that? The next neuron. There's no touching. They're very close, but they don't touch. Do you see that? Even at the effector organ. Now, that is because, that's called a synapse, that gap. And that is because the neuron comes along and there is a gap between it and the next neuron or the muscle. All right? So what it does is, it synthesizes and makes neurotransmitter. And the neurotransmitter is a chemical. And that chemical then binds to the next one. So the message is not in any way compromised. That's a chemical that's carrying the message from one neuron to the next one. So that chemical is called a neurotransmitter. That gap is called a synapse, right? The neurotransmitter is synthesized in the previous neuron, and that one is called an axon. Okay. Now, what is reuptake? Reuptake is a process by which chemicals in the brain called neurotransmitters are absorbed back into the transmitting neuron after they have been released into the synapse. This controls the intensity and duration of the signal being sent. What it's actually saying is that when you synthesize neurotransmitter, you don't use all of it. So the neurotransmitter just doesn't float around there, it gets taken up again. And this is important, and I'll tell you why. So when a chemical signal is being transmitted from one neuron to another, the first neuron releases the neurotransmitter into the space between the neurons. That's the synaptic space. Once the second neuron has received the signal, any additional neurotransmitter that is left in the synapse can be reabsorbed. So only a certain amount of the chemical is used. Now, dopamine, serotonin, norepinephrine are neurotransmitters that we've known about for decades and decades in our brain. The neurophysiological mechanisms responsible for the psychological observations of emotions and motivated behavior, although the neurotransmitters dopamine, serotonin, and norepinephrine have been implicated in our behavior. Now, serotonin is a complex structure, but what I wanted to show you is, look what it's made out of. Look what it's made out of. Tryptophan. What is tryptophan? An amino acid. A building block of protein. It's all related, the body. It's absolutely amazing that the more you study the body, you see all these interlinks. Everything somehow or the other is connected. Okay? Now, serotonin is regarded as a chemical that is responsible for maintaining mood balance, and that a deficit of serotonin leads to depression, sadness, feeling down, having a loss of interest or pleasure in daily activities. These are symptoms of depression familiar to all of us. So now I don't want to say very much more about serotonin, except that the most common class of drugs for the treatment of these conditions like depression. So if a doctor says, or a psychiatrist says, I think your serotonin levels are low, they'll give you fluoxetine, Prozac, which is a happy drug, right? Okay. Okay, now, you know about serotonin. Prozac makes you happy when you've got too little serotonin. Look at the brain. The brain has these regions where dopamine is made. Dopamine is a very important neurotransmitter. You've got a shortage of dopamine, you can get Parkinson's disease. Right? Now, look at the circuits where serotonin is secreted. So, you've got functions, dopamine's functions are reward, motivation, pleasure and euphoria. The motive function is fine-tuning, compulsion, and helping you persevere. Serotonin helps your mood, helps your memory, helps you sleep, and helps you think. So they're particularly important. But now look at it. Look at dopamine. You think it's all in the brain. But this new field is telling us that the intestine produces 50% of your dopamine. Can you imagine? So this gut-brain axis is a very busy highway. Right? Now, some gut microbes, example E. coli, produce 10 to 100 times levels of dopamine seen in human blood. L-DOPA is used to treat Parkinson's disease, a debilitating disease. Okay? Now, serotonin. The intestine produces 95% of all the serotonin in the body. So it was never in the brain. What you're seeing here has probably come from the gut. Okay? Gut microbes produce factors that can mimic or block serotonin production and or action. Deficiency can cause depression. Now, I must mention one of my favorite neuroscientists. How many of you watch Charlie Rose's show on TV? He once interviewed Eric Kandel, the American neuropsychiatrist. He was a recipient of the Nobel Prize in Physiology or Medicine for his research on the physiological basis of memory storage in neurons, and he used the axon of the squid because it's so big and thick and it's easy to work with. Kandel has had quite a life. During the war in Germany, they had to leave Vienna. And he said at one interview, and that struck me, it's okay to be sad, but too much sadness for too long is not normal. It's a depression. That's what he told Charlie Rose. And my daughter and I have had the pleasure of reading first-hand accounts by psychiatrists on their own mental problems. There was Claire Redfield Jamison, who wrote about her own bipolar disorder. She was at Harvard. There was Ellen R. Sachs. She wrote about depression. And who was the third one, Sumaya, the one in California? Can you remember? So there was a third one, a woman who said that they tied me down to my bed. And she was a doctor, she was a psychiatrist, she was schizophrenic, she knew it, but she said they had absolutely misunderstood her. And they just kept her tied as if she was a criminal. When she was trying to express herself and say, look, I know what I have, but I'm not a danger to you. It's all very sad. And this is the problem with modern day psychiatry, I think, where the psychiatrists feel that they're not really getting to the patient. They're not really getting to the patient, and they should actually allow the patient more space to say exactly how she feels, why she feels that way, and try and understand her better, rather than actually imposing a diagnosis and say, you have bipolar or you have schizophrenia. Okay, so this Prozac is an antidepressant, right? Now, belonging to the selective serotonin reuptake inhibitor class of drugs. Right, now, just as I explained to you, when you secrete the serotonin, it goes to the next neuron, but there's too much of it lying around, and so the first neuron which secreted it reuptakes it. Right? What Prozac does is it prevents that reuptake. So it's a serotonin reuptake inhibitor. So eventually you've got more serotonin there. That's why you get happy. Okay? Now, or commit suicide. I mean, there are problems with that, I know, with the drug. Gut bacteria and mind control. I was once asked to take Prozac because of my obsessive compulsive behavior. And I thought, no. Gut bacteria and mind control to fix your brain, fix your gut. This is Simon Carding, who once swallowed a pill can, which had a sort of, which could take a video of his gut. Some of them go to great extents. So he's a professor at Norwich, and he swallows a tiny camera to video his gut. And he gave his talk on the gut-brain axis, fix your gut, fix your brain. I think he's the world's leading expert on this. And he asks questions. Now, this, ladies and gentlemen, is from his talk, and I could stop here right now and say to you, I'll give you the website address, and you can go home and listen to the talk on your screen. But do you mind if I just carry on for a little while, just to cover the main points? Do you think? Okay. Genes and lifestyle. You think genes and lifestyle cause disease? They could keep us healthy or cause us to be ill, right? Steven Pinker, in How the Mind Works, if you read his book, he says, genes are everything. They make you and they eventually kill you. Now, there's been a whole debate by experts around the world about what a gene does, how it reacts with the environment. And my favorite sociobiologist, Edward Wilson, he was opposed by another fantastic scientist, the late Stephen Jay Gould. So they talk about a gene culture co-evolution. It's not just genes alone. You have the genes, they design your body, but they don't just sit there, they interact with the environment. And so the final product, your final behavior, is through an interaction between your genes and your environment. Okay? Now, microbiota have come into the picture. So since 2000, a fantastic leap in publications because of technology. Microbiota have come in between our genes and our environmental factors. They are now seem to be the mediators of behavior in between the gene and the environment. They influence our gene and they respond to what we eat. So now, at one time, there was a problem. We couldn't grow these microbes in a dish, in a tissue culture in the laboratory, because they can't all be cultured, because they die very quickly. But now we have gene sequencing. So we need a little amount, and you can define all the microbiota in your gut, okay? But we haven't quite got there yet. What we eat influences our microbial populations in our guts, okay? Now, there is even a link between the microbiota and autism spectrum disorder. There's evidence of causation. So they have a mouse model that's autistic, very, very obsessive, scratching itself. So what they did with this mouse is they gave it some probiotic treatment. It altered the composition of the gut microbiota. It improved the epithelial barrier integrity. What they found in this autistic mice is that their gut cells were not tightly adjoining one another. The gut was leaky, and that's not a good thing because stuff gets into your bloodstream and causes an infection. They think that's one of the characteristics of autism. Okay, so this probiotic improved epithelial barrier integrity, reduced the leakage of particular GI metabolites. They didn't leak into the blood. It restored the serum metabolites, and it ameliorated specific autism-related behavioral abnormalities. I think this is absolutely fantastic because with autism, I've been to a lot of talks on autism, and even here in Cape Town, and everybody focuses on the brain. They say it's a fault in your wiring or something to do with the brain. And one day I did ask at Falkenberg at a talk, is there any other thing about the autistic individual that you think is worth investigating? They said, no, it's all in the brain. But this is new. This is new. Okay? So gut microbes are essential for health and survival. Now, in the lab, sometimes immunologists use germ-free animals. So they can actually breed a mouse or a rat that is completely devoid of germs. Germ-free animals have nutritional difficulties, poor growth but long life, defective gut and a leaky barrier, defective immune system. Introduce one pathogen and they die. So while you're sitting here, you're absorbing so many pathogens. Fortunately, I don't see any of you dying. So your immune system is working very, very well. So if you in germ-free animals, you have defective antibody production and they're susceptible to fatal infection. They have an altered neurochemical production and brain development. That means they have no bacteria in the gut. So their brain development is retarded. Your microbiota, each one of you, is unique like an individual fingerprint. You're going to tell me when to stop, right?
What else in common?
Oh, it's not even an hour. Okay, I'll carry on. You tell me when to stop. So your microbiota is unique. It's unique like an individual fingerprint. The microbiota of family members are more similar than unrelated individuals. So environmental factors do play a role. Microbiota of monozygotic twins has similar variability as that of dizygotic twins. Early environmental exposure is the key determinant of adult microbiota. Genes and who we are is nature, and that's important, but so is how we live, which is nurture. But we all, and this is from our genetic revolution, share a core microbiota. So although there's variation, we share a core microbiota. It's independent of gender, race, age, and geography. I wish I knew this during apartheid, I would have mentioned it. 75% of the species are common to more than 50% of the individuals. 57 species are common to more than 90% of the individuals. Two types which are conserved because they perform important functions predominate in all individuals, and those two are bacterio-DTs for energy production, amino acid transport and carbohydrate metabolism, and firmicutes for carbohydrate and vitamin metabolism. Okay? So, a functional core of microbial species is highly conserved in everyone for the degradation of complex carbohydrates, synthesis of essential fatty acids, sources of essential amino acids, and vitamins which we cannot produce in the body. Source of microbial species. Now, mothers give their microbiota to their children. The baby is exposed in the vaginal canal of the mum during birth. Then there's a postnatal exposure, but the baby is spending all its time with the mother. Those who have cesarean sections could result in exposure to other sources, like the staff who are delivering the baby. Their microbiota could be transferred to the baby, and that has implications of health in adulthood. Microbiota also are determined by the home, hygiene, ward environment, breast or bottle feed, water, food, pets, geography, salivary exchange, intimacy, grooming. They all affect your microbiota population. By the age of 3, most of your microbiota has settled in. But bacterial populations can change in an individual as we age, frequency of antibiotic use, and the anti-cancer drug tamoxifen and the blood pressure drug minoxidil can increase the toxicity of some of your bacteria. NSAIDs, non-steroidal anti-inflammatory drugs, and sulfasalazine, which we use for the treatment of Crohn's disease, inflammatory bowel disease, protect against harmful species. Bacteria in turn can produce enzymes and alter the structure of some drugs and prevent their mode of action. Choosing drugs based on an individual's gut microbes is a real possibility. We are entering the age of personalized medicine. Your gut microbes influence what and when you eat and whether you remain healthy or unhealthy. And Simon Carding has a hypothesis or a prediction. He says the makeup of the bacteria in the gut is selected to a large extent by the food we eat and the behavior in our own environment. In turn, the bacteria can, by influencing how our body works, influence our appetites and food preferences. Migrating populations show the importance of diet in the makeup of the microbiota. Fasting reduces microbial diversity and feeding increases it. Probiotics. Probiotic bacteria commonly found in yogurt and produce the neurotransmitter GABA. This is your area, not mine. GABA binds to overexcited neurons and reduces the activity, thus reducing stress. GABA receptors are targets for sedatives, benzodiazepines, ethanol, muscle relaxants, and anesthetics. Obesity. When they took poo from twins, one fat, one thin, the bacteria were fed to 2 groups of rat. The bacteria from the fat, a fat twin, made the mice fat, and the bacteria from the thin twin didn't make the mice fat. Now, fast food kills